Good afternoon, everybody. Welcome to the 14th Annual Shishgal Lecture. We're pleased and proud to have Dr. Dave Chung visiting us from Vanderbilt University, and he'll be giving us a grand round today on targeted therapies against neuroblastoma. I should acknowledge Dr. Shishgal was a pediatric surgeon in the DC area for many years, and also acknowledge that Dr. Larry Cohen, a good colleague and friend of his, has been very benevolent to support this lecture over the years. Dr. Shishgal was the go-to pediatric surgeon based mostly out of Holy Cross, but operated all throughout the area and brought complex cases here to Children's. Dr. Chung comes from Vanderbilt. He trained at the University of Texas Medical Branch in Galveston for both med school and residency. I first met him in Cincinnati, where he was a pediatric surgery fellow 1994 to 96, and we did a lot of ECMO together. He's since moved on, done great things. Spent a couple of years at the University of Alabama, Birmingham. Maybe that's why I got recruited there, but when I went, he was gone. And then he spent some time back in Galveston, and he's currently now the professor and chairman of the Department of Surgery at Vanderbilt University. He spent many, many years researching neuroblastoma. He's had continual NIH funding since 2003, and he's also written over 160 papers and has mentored many, many lab residents and colleagues throughout the years. And I'm honored to call him a friend and colleague, and looking forward to this lecture today. And this has been great so far. Thank you.
Good afternoon. Thank you very much, Tim, for those kind words. It's been a little over 20 years now since we've all sort of struggled in the bedside in NICU, cannulating some difficult babies, and it's great to see you doing so well. And so many friends and colleagues, Dr. Newman, Sandler, although I visited once here during interview trail for fellowship, 91. This is my sort of second time. I have to say I recognize some aspect, but boy, this hospital has just exploded. It's great to see such a phenomenal hospital and number of outstanding programs. So I'm really thankful and grateful and privileged to be here to share some of our work for the past little over a decade and a half, but I won't give you the entire story. I will. Just gonna do I just press this or just sorry, takes time, too complicated. Oh, thank you. Oh, that'd be great. Thank you very much. I know Tim mentioned about Dr. Shishgal, and as a visiting lecturer, I think it's always good to learn and know about what the endowed lectureship is all about. So I had a chance a little bit about his outstanding academic records and accolades, and obviously he's a grad of Princeton University and received his medical education at NYU. There he did the residency at the Bellevue internship and a residency, which is incredibly difficult and challenging hospital to work in. After finishing his fellowship in Detroit, he's returned to his home area and practiced for over 2.5 decades. What I've read and what really struck out the most in my learning about his legacy was that he was clearly known as an incredible clinician, having received the Physician of the Year as well as Clinician of the Year by really very well recognized by the community and what he's contributed to care of children as well as working with colleagues. And one of the sort of credos that I discovered learning about Dr. Shishgal was that he stood by this, that early in his practice that he decided that no child operative discomfort would go for naught, but that each child's surgery would be woven into a broader fabric of experience. I think that's just sort of a very important message. And it sort of kind of resonated with me because I sort of asked to come here and talk about my research and what I do. And I certainly wanted to be able to kind of understand that background. And today I'm going to share with you a bit about what we have undertaken for the past almost couple decades of trying to better understand neuroblastoma and trying to come up with better strategies to manage and help these kids succumbing to this devastating cancer. You all are pediatric specialists, so I don't need to delve a whole lot into this background slide. We all know how challenging this cancer is for our infants and children. Despite incredible advances in both basic and translational research arenas, I think if you look at an overall survival curve of all pediatric cancers for the past several decades, I think that you would agree there has been some, but not enough of improvement. So there are plenty more, or a lot more discoveries to be made to make some impact for these unfortunate infants and children. And certainly, as we all have experienced, this is not, you know, the most pleasant sort of sight when we're asked to see whether on the wards in that emergency room. All right, so what is neuroblastoma? Again, classically described as small blue round cells. Neoplasm of childhood, we're quite familiar with seeing these very poorly differentiated, derived from neuroblast cells. Rather than just following its proper lineage, whether they remain undifferentiated or revert back to dedifferentiated state, exact mechanism clearly obviously is unknown. And some of the recent years of stem cell work has delving into better understanding these proper sort of lineage cell fate for neuroblastoma and how it may play a role, and perhaps that will have some more sort of insight in upcoming near future. I do have to sort of acknowledge my fellowship mentor, Maury Ziegler. Many, if not all of you, may know him. Maury Ziegler was surgeon-in-chief back in mid to sort of late 90s in Cincinnati, where Tim mentioned that I was a pediatric surgery fellow. It was an incredible experience, and there are some really kind of key moments during 2-year fellowships of staying up many nights. Not really, you know, we never stayed up nights. But I remember oftentimes in his chief rounds, he would ask to a group of fellows and residents and students that, you know, what do neuroblastoma and melanoma have in common? Of course, as a trained general surgeon, we're familiar with managing some patients with melanoma, and he would always challenge us with that sort of teaching moments. And he would elaborate that both are neurocrest cell-derived malignancies and some can regress spontaneously, and which I really honestly didn't know a whole lot about neuroblastoma at the time, nor actually melanoma, just enough to know how to manage it surgically, so in order to pass the certifying exam. But I thought that was really interesting. How can these, like, very, very aggressive, clinically devastating cancers can just themselves spontaneously regress? I mean, I thought, wow, if one can figure out, that was my aha moment, one can figure out the mechanisms that may be involved in those kind of tumor regression, perhaps that could be a really impactful novel therapy or discoveries for neuroblastoma. And so hence, after fellowship, a brief stint at UAB, couldn't get myself out of water to even go to the bench. So I had to go back to Texas to start my basic science career, along with establishing a pediatric surgical career. All right, you guys all know this. When we talk about neuroblastoma, it's often categorized or discussed in terms of risk-based outcomes. So as you see in this sort of very sort of simple survival curve, when you analyze their overall, whether it be a 2 or 5 year outcome, we often like to sort of reference to their risk-based classification. So if you look at this clearly, as shown in red circle, babies or children in falling into high-risk category clearly do poorly. And there are a number of speculations and potential reasons for that. Some are known and some yet to be discovered. So when you look at neuroblastoma investigators or studies in tumor biology, current clinical problem is that we don't know and we're not doing any better to really help these kiddos in high-risk categories. So again, if you look at over several or past couple decades, that it has improved somewhat over the course of since the early 90s, where with all the advances, all the treatment strategies, that we are having a little better event-free and overall survival, and shown by these blue arrows. But how can we get to near flat where we can save or salvage and help all these patients? That is obviously the big holy grail. These are some listing of pre-relapse treatment protocols that many of you administer in this room, including surgery, what have you, including immunotherapy and retinoid differentiation agent therapy. These pretty much have become standard. Despite all that, it's sort of fair to say that we're making very little index. So this is sort of a gross picture. Drs. Kane, Newman, Chandler, you've all managed these tumor resections, dozens, if not hundreds. Some are relatively straightforward, some very, very challenging. So obviously this is something that we at the clinical bedside are still struggling to do more or do better. So today I just want to kind of maybe share a little bit of some of our work, some of the recent work that I'll just mention about targeting MYC. This has been a holy grail of neuroblastoma biologists, trying to figure out how to target and effectively dysregulate MYC, which is obviously well-known oncogene for this cancer type. And then what about the metastatic disease? We often not only at presentation, but relapse, these kids will show up with disseminated disease. How can we take care of them better? And then with that, along that relapse and high-risk tumor, I want to just mention a brief about last year or so our laboratory's work on epithelial-mesenchymal transition and what it means for neuroblastoma, and can we, with our data, introduce or develop new targeted therapy. So that's sort of a brief outline of what I'm going to talk for the next about 20 minutes. All right, MYC and oncogene, well known, 30+%, especially in high-risk tumors, are amplified. And easy to say that it's equivalent or it relates to translating to very poor prognosis. Now, Bill Weiss has developed and published this in the late 90s of transgenic MYC mice, and there was an exciting buzz and a lot of studies. There still are, but it's fair to say it was a very difficult model to work with, and there's been additional transgenic lines that we work in various laboratories, but certainly hasn't translated into a major, impactful, meaningful new therapy for our kids. There are many well-known downstream effectors that are known to be involved in regulation of tumor, whether it be p53 or a kinase, NDN2. This is just a little brief sort of 3 examples of the downstream targets that other laboratories are currently in active investigation. But it certainly is sort of fair to say that no significant clinical success in targeted therapy against MYC has been developed. So then, hence the question, MYC is it really a sort of undruggable target in neuroblastoma? Some would say yes, and others perhaps would strongly argue, and hence we move forward with our studies. I personally, in our laboratory, have been interested in some of those newer, for the past decade, compounds that have been known to target MYC. I'll just give you a little background on the BET inhibitor that we've worked with in JQ1. It's been published in a number of high-impact journals the last several years. BET, or bromodomain extraterminal proteins, are known as epigenetic regulators. There's been a lot of interest in cancer biology about better understanding the role of epigenetic regulation. They're known readers of the acetylation process of the lysine residue of the histone. So there's well-known commercially available compound inhibitor of BET-JQ1, which is known to regulate MYC in a variety of cancer. And furthermore, in neuroblastoma, it's been shown to regulate this MYC or MYC and hence cell cycle and apoptosis. So we've done extensive work in that area, and certainly I think we're at the sort of scratching the surface. And as you may all know, that MYC has been also known to be critical in regulating differentiation process of neuroblastoma, hence the retinoic acid and other agents are sort of routinely used. So we had hypothesized that perhaps JQ1 also impacts or regulates tumor progression in neuroblastoma via differentiation, and there's plenty of evidence that we've been able to show or publish that it does indeed have a role. Second, metastases. This sort of showing a little liver nodules in a child with the right adrenal primary. This is often a problem, a dilemma, whether patient would sort of be a resectable candidate or not. Clearly, would go through the induction therapy to see if we can eradicate the metastatic lesions before proceeding with the gross resection. I'll just kind of revert back to 20-plus years ago when I was a surgery resident in Texas, as a surgical research fellow, studying many of the molecular work at that time. One of those compounds or peptides that I was introduced by my mentors in general surgery oncology world was Bombesin, or that's the mammalian equivalent of a gastrin-releasing peptide. So I'd done some studies back when I was a surgical research fellow, but then just the same time, Bob Solomon, whom many of the surgical colleagues in this room are quite aware, Surgeon-in-Chief at Seattle Children's Hospital, had done some work where he had shown and presented that gastrin-releasing peptide is an autocrine growth factor for human neuroblastoma. By analyzing various paraffin-embedded blocks and some simple in vitro assays in the early 90s, he subsequently published this as titled Secretion of the GRP by Retroperitoneal Tumor. He incorporated some of the other sarcomas and other tumors in addition to neuroblastoma that they indeed secrete. He was able to measure, and it was sort of an interest certainly to me as a sort of a young wannabe surgical investigator, having had some exposure to gut peptides and its relevance in neuroblastoma. So then, now moving back to sort of more recent times, establishing a lab model with regards to better understanding neuroblastoma metastases or molecular mechanisms, I set on to sort of introduce splenic injection of human neuroblastoma cells to see whether I can establish liver metastases so that I can harvest subsequently and better understand at the molecular level. So this is where I had actually transduced or knocked down GRP receptors in human neuroblastoma cells, and then subsequently injected into a murine model in the spleen to see what would they do to compare to the control normally expressing GRP receptor human neuroblastoma. So as shown here in the photograph, when you silence this receptor for this particular peptide, you can see much less or almost no liver metastases compared to control. And we validated that with histology evaluation, where these are truly small round blue cells of neuroblastoma. So that kind of gave us a lot of sort of ideas and subsequent studies that GRP peptide certainly plays a role in metastases in neuroblastoma. More recently, I wanted to really better understand these highly aggressive subclones of high metastatic potential neuroblastoma. So of late, what we've done was to sort of inject and establish liver mets of human neuroblastoma, labeled here as LM1, harvest them, reestablish their tissue culture, and then re-inject, so enhance, and they're highly selected, very aggressive subclone, and we've characterized it with a lot of in vitro studies, and we're able to see their metastatic potential is enhanced. So we followed up with then analyzing with various molecular techniques what exact mediators. Some are inflammatory mediators, others are more about growth promoting factors in the GI tract, and we've done some subsequent studies to sort of identify them in conducting much more sort of targeted therapy, new strategies that we're testing out in vivo. Now I want to shift into what I've been really, really interested in for the past year or so. That is the concept or process of EMT. That is epithelial-to-mesenchymal transition, well established in adult cancer literature, to be associated with cancer invasion, metastases, and drug resistance. So shown in this diagram, as you see from the left to right-hand side, there are clearly well-established markers of the epithelial cell, such as shown in red box, E-cad, as well as occlusion. And when the cells go through this EMT process or transition, they develop this sort of a less polarized, more lumpy, and this is where we equate to having higher invasive potential. This is sort of what, in a very simplified explanation of what cancer cell potential or changes in the cell's increased potential from metastasis or tumor invasion is sort of thought to occur with regards to EMT. So this is, again, well sort of established inflammation in adult cancer, not as much so in neuroblastoma. So as shown in this slide, and referenced here by Guinea and all work, in colorectal cancer, abbreviated here CRC, this group was able to identify the EMT subtype that are responsible for disease or colorectal cancer recurrence and poor survival. And they labeled it here as CMS4. And they've analyzed that with their overall patient disease recurrence as well as survival. And clearly, there was an association with expression of the EMT markers and poor survival. So that's, again, in colorectal as well as lung cancer. So well-known established cancer biology information. The initial question was, can we identify small molecule probes that reverse EMT and restore E-cad, which is a marker for epithelial phenotype expression? Now, this is sort of work done by a couple of the colleagues at Vanderbilt, my chair, and Beecham, who's worked in colorectal cancer, spans over 2 decades. His primary focus had been TGF-beta in colon cancer progression. But he and working with Lindsay, who's a chemist, were really interested in developing a new targeted therapy for colorectal cancer. With that initial question, then the hypothesis was that can small molecule probes that inhibit or reverse EMT process can provide further insight into biology of EMT and hence essentially target this for therapeutic purposes. All right, so this is sort of, again, working with Vanderbilt Institute for Chemical Biology. It was a high throughput screening, a drug screening process based on phenotype-based small molecule screening. There was initially over a library of 80,000 small molecules, and we used in-cell Western blotting as a sort of readout to see what molecules will actually induce expression of E-cad, which is a marker of epithelial phenotype. So initially, there were about 1,600 hits. Then after multiple screening, exactly 4 passes, narrowed down to 30 hits, and then they were down to 13 hits based on dose response study. So that was a start. And then we analyzed, our colleagues, that really this process showed that the most important chemical biology structure was the Isoxazole-based backbone. And that's hence then really started to further develop new compounds with similar structures to test out in the sort of readout. And there were over about 400 new drugs or molecules synthesized and tested. And just to give you kind of a brief result, a compound labeled ML327 was the most potent and specific inducer of E-cad expression in colorectal cancer. Hence, that was the drug that we subsequently used for further analysis. And this is, next result is simply just to validate in vitro findings of indeed this compound ML327 reversed the EMT process in blocking invasion or invasive potential in colorectal cancer. Right, left-hand panel showing some fluorescent staining with TGF-beta treated cell. When you co-treat with ML, you can actually induce E-cad expression, and showing as emerge, it indeed validates expressions are indeed localized to those cells. And then this is the invasion assay was shown that is indeed effective, showing the phenotype change with the ML327. So just to summarize those few experiments, we found that EMT is associated with metastases and that therapeutic resistance and poor outcomes in epithelial cancers, and identified the Isoxazole-based family for high-throughput screening, screening over 80,000 small molecules. And the structure-activity relationship eventually identified this compound we call ML327 as a potential most potent inducer of E-cadherin expression in colorectal cancer. So hence, as this compound reverses EMT, blocks the invasion of the colorectal cancer cells. All right, then extrapolating that to cancer of our interest, neuroblastoma. Well, what do we know about neuroblastoma in EMT? Not a whole lot. I mean, there are some information published in the literature. Neuroblastomas are obviously posted to rise from neuropressed cells, which gain migratory potential by undergoing EMT transition or process. EMT changes are observed in early neuropressed migration, parallels that of carcinoma or cancer metastasis. So then mesenchymal features have been well known or have been associated with resistance to both chemotherapeutic as well as targeted strategy. So we raise the question, is a reversal of EMT a viable therapeutic strategy for neuroblastoma treatment? We hypothesize that ML327 may be used as a chemical probe to reverse the epithelial-mesenchymal transition and to evaluate its effects on neuroblastoma tumors. First, we wanted to demonstrate what is the sort of basal expression of markers that represent epithelial versus mesenchymal cells. So shown here in black bars is CDH2, which is a gene name for NCAD. Again, this is representing more of a mesenchymal. There's abundant sort of area expression in sort of multiple cell lines, whether MICAN or non-amplified cell lines that we tested. Conversely, when you look for the epithelial markers shown here in open bar CDH1, which represents actually E-cadherin, there's hardly any detectable basal expression. When you look at COCAC R2 database, it's well established that lower expression of CDH1, again, reflecting the epithelial marker of E-cadherin, is associated with neuroblastoma recurrence and overall worse or higher mortality in kids and children, as shown in the red line. So we hypothesize that ML327 may be used as a chemical probe, and we set out to do the test in vitro as well as in vivo. First, we wanted to see whether ML327 will actually induce this mesenchymal to epithelial transition in neuroblastoma. So by measuring what this drug does to our cell line, as shown here in open bars, when you treat these cells with ML327, you can see the expression of E-cadherin as well as occlusion, so two representative markers of epithelial phenotype. We are able to show morphologic change, shown here compared to the left column of a sample of pictures. With ML327 treatment, you can see much more elongated spindle epithelial phenotype as versus control treated vehicle. When you do a RNA-seq analysis and determine various markers of epithelial, you can see a peak with the treatment with ML327. So those are all kind of correlating, lining up to the fact that, yes, indeed, this compound may have some significant changes or effects in neuroblastoma. So then we set out to determine some various simple growth and viability and soft agar assays. Shown first in panel A is the simple cell colony counts with or without ML327. It shows here in multiple cell lines, again, both MICAN, non-amplified as well as amplified. This ML327 reduces cell viability or cell growth. Shown here with cell viability assay, significantly attenuated. Panel C is a soft agar, sort of representing a in vitro sort of a representation of the tumor potential. You can see a significant attenuation of the colony growth. And when you do a cell cycle analysis, you can see an increase in G1 phase, suggesting that indeed maybe ML327 may induce cell differentiation. We, again, followed up with RNA sequencing analysis and showed that ML327 shows an increase in differentiation signature or markers of genes, so all correlated with our in vitro growth findings. We then wanted to see actually what would happen if we treat and wait for several days. This particular experiment was over a course of a week. Shown here on the left-hand side of a graph or bar, soft agar colonies with treatment with ML. 7 days later, you can see a significant decrease in colony growth. And then when you measure the sphere-forming potential, which is one of the sort of tools that my postdoc developed in our laboratory, it significantly attenuates this effect. All right, then with those in vivo or in vitro findings, we went to the vivo murine model to test. Obviously, those are sort of stepwise progression of your study before you can have, introduced to possible translational as a new drug. This is a neuroblastoma, human neuroblastoma cells, treated in vitro with ML. And then we harvested and inoculated into the subcutaneous tissue plane of nude mice. So this is what we would think that they're pre-treated to induce epithelial phenotype or expression of E-cadherin. So when you do that and inject, it's shown in sort of a flank. You can see the control is in the where the tumors grow, but the left shoulder flank is where the cells injected, pre-treated with ML327. So they really weren't able to even establish the xenograft. Hence, we speculated or we concluded that ML327 actually does also block tumor-initiating capacity of neuroblastoma. And I think it sort of shows the tumor eventual volume change in the bar graph. Well, what would happen if you start treating this compound, or treat with this compound, in a murine model of xenograft that have neuroblastoma established? And we're not talking about pre-treatment, so we have the xenograft randomized treatment control group and start treating them over the course of 2+ weeks. It's shown in this sort of line graph compared to the control. ML327 also was effectively able to abrogate or decrease the size of the tumor growth of an established MIC-and amplified xenograft in our hands. So it shows at the time of harvest, weight correlating nicely with the volume measurement throughout the experiment study. So with that, we were able to sort of find that ML327 is indeed capable of inducing epithelial-like features in neuroblastoma in vitro, and also induces growth arrest and gene expression changes consistent with the induction of differentiation. And IP administration in vivo model of ML327 is capable of suppressing tumor growth of established xenograft of neuroblastoma. So really the million-dollar question is, what is the direct target? What is the molecular mechanism? And that's where we're at in our laboratory at this juncture. This certainly hasn't been an easy challenge. We've met many failures and continue to do so, seems like every day. We sort of took on a couple of strategies. It's just to go to show you some of the younger members of the audience. Research is not easy. I would say easily, Dr. Sandler would agree, 90+% of studies are failures or fail. I think you really just have to have some perseverance and continue to move forward. First aspect of the strategy was, can we sort of look at it from a chemical biology aspect? That is, can we tag this compound to see what are intracellular direct targets of this neuroblastoma? So as listed, just sort of simple sort of strategy that we use. We're able to tag the compound, but in the end, it didn't work because it sort of we eventually lost the tumor inhibiting efficacy when we tested it out. So that didn't work. And then we have turned into RNA sequencing to see if there's some gene signatures or genes that we can identify to further sort of figure out what is the molecular mechanism. And indeed, when we did that, we have negative correlation with MYC gene, shown here in this sequencing signature map. We were encouraged. Obviously, MYC, as I said it multiple times, an important oncogene for neuroblastoma. Was able to, using some simple Western, show that in multiple neuroblastoma cell lines, with the treatment with ML327, both the c-MYC and MYC protein expression was decreased. And you can show in panel B, there's a time course showing a nice kinetics of induction of the decrease in MYC expression with the treatment with ML327. So then we're now currently at looking at a little more at the transcriptional level. How does this really regulate MYC? And top panel showing some Western that with the treatment with the cyclohexamide, that protein levels of MYC or MYC wasn't altered with ML327. So we said, okay, maybe it's not the translational process. Let's go to the transcription. Shown here, the mRNAs decreased by, levels decreased by ml. But when you actually treat with the actinomycin D shown in the right-hand panel, their levels did not change, meaning there was no really mRNA stability issue. So we're now sort of moving to really better understand how ML327 may be regulating MYC message at the transcriptional level. So it's just a little snippets of kind of what we're actively doing. So to summarize, so far what we've done in the last recent couple of years, that our chemical probe here, labeled as ML327, is capable of inducing epithelial-like state in neuroblastoma in vitro, and it abrogates neuroblastoma growth in vivo by promoting differentiation. And we're able to show that ML27 blocks xenograft growth in mice, but the direct mechanism of action for this compound remains unknown and is sort of a subject of our ongoing studies. But we believe that the isoxazole-based family of compounds certainly may be useful, lead point to the development of novel targeted therapies against high-risk neuroblastoma. So that's what I wanted to share with you. I know I sort of showed you snippets of different aspects of our lab. Clearly, without these individuals in the lab, we wouldn't be able to sort of generate this kind of a work. I think Eric Rellinger, shown here with Beer, is a really talented surgery resident. He's in the match this year, and you may see him. Collaboration with Beecham Laboratory and Lindsay Lab was about saying incredible things to them and our funding sources. So I hope that kind of sheds some little bit of insight as to at least what we are doing at Vanderbilt with regard to neuroblastoma studies, and hope that in the near future that we can translate into a new novel therapy. Thank you. Yes, please.
It's a really elegant story, very clearly told. I congratulate you. Thank you for sharing it. I have a question that actually goes pretty far upstream in this story a little bit. You may know that the Children's National has a Rare Disease Institute. We're very interested in the early detection and management of a range of rare diseases. And a lot of that is dependent on early life screening and the newborn screen. I have a crazy question. Is there any way to screen the population for MYC enhanced expression? Has anyone ever thought of doing that before to detect that rare subset of children who are going to go on to develop this devastating malignancy?
No, that was an excellent question. I mean, sure, I don't know of any. Obviously, closest thing to screening for neuroblastoma is that obviously much more so in Japanese investigators a couple decades ago screened through urine metabolites to identify some asymptomatic infants that may have these tumors. And long short of it, I think even their aggressive approach to resecting these really, I think they were mostly resecting differentiated or ganglion neuromas or more of a benign phenotype of neuroblastoma. I think certainly in the States, we've not done that. As you all know, the infantile neuroblastoma do occur, and studied by our colleagues at Induction through the help of the COG have sort of introduced this guideline of monitoring, because many can regress and safely monitor. And if tumor growth doesn't occur, then without really harm, that we can actually still proceed with the resection. So those are some examples of kind of what we know with infantile or very, very early stages of neuroblastoma. But with regards to screening for MYC or MYC family of genes or protein, I don't know of any. I'm not sure whether, that's a very great idea. You know, I have to put some sort of thoughts into it, and perhaps we could, because I mean certainly these tumors do occur in utero. But no, short answer to your question, I don't know of any, but certainly we're looking into. Yes.
Distinguish the mesenchymal versus the epithelial. Have you found or studied any other markers to discern whether the differentiation did or did not occur?
Yes, thanks for that question. Yeah, we've pretty well established using various differentiation markers, whether that's neuron-specific enolase or others that are well established in our field, to show that the differentiation process is affected by this compound. But with regards to just determining reversal process of EMT, that is mesenchymal to epithelial transition, we've only really used those two, E-cadherin and Recurrence, and there are a whole host of others developed. They're just most abundantly or most commonly used markers, and we wanted to stick with that to be able to validate, because it certainly was a sort of new information for neuroblastoma biology. But yes, to answer to your question, to determine the degree of differentiation this compound influences, we use NSCs and other markers to validate.
Hi, I'm Jeff Dome. I'm the Chief of Oncology. Thanks for the lecture. Very nice. I have a question. As you pointed out, we use retinoic acid as a differentiating agent, so that has already made its way into practice. Do you see a different response in the cells in response to retinoic acid treatment compared to your drug? So would retinoic acid have the same effect on MYC amplification or expression and E-cadherin expression? So is this, in other words, is this specific to your drug, or are these effects a generalizable effect to a differentiating agent?
Thank you for that question. I know my colleague Bo Lavorne works closely with you in Wilms tumor. He talks awfully highly of you often. I don't know whether retinoic acid mechanism is exactly the same as ML. We clearly know that retinoic acid does not induce E-cadherin and Recurrence protein expression. They certainly do very similar things to neuroblastoma. In the end, they're kind of, could we work both sort of using it synergistically to further enhance the differentiation process that perhaps retinoic acid is in this ML compound? Well, what we've done is really using a cell cycle analysis to get a readout of differentiation and measuring some of the protein changes. So I believe that retinoic acid's effects on differentiation neuroblastoma and ML compound works in a different mechanism, but perhaps in hope that they could work together to potentiate synergistically is kind of what we're hoping. But whether they're actually doing it or retinoic acid works in the same way, I don't believe so, based on our at least preliminary data.
Dave, thank you very much. It was excellent work. I have a couple of questions, but I won't bore everyone with them, and we can chat privately. One of the congratulations on really trying to find new drugs for an old disease, and I think that's excellent. one of the questions I had is when you gave your ML327 compound and the tumors grew at different rates, did you see a difference in the phenotype of whether they were mesenchymal or epithelial as opposed to controls? Because they both grew tumors, and to 24 days is very early. Did you see any changes that you were actually impacting the phenotype?
Great question. As we all kind of share our research advances, you know, oftentimes that we show the sort of good results, and many aspects there were completely puzzled. So, direct answer to your question, we're not able to see any differential effects in mesenchymal versus other components of these tumors in our in vivo model. I think that certainly is a major criticism, because even though we have in vitro data to all suggest that's what's going on, in vivo, we're not sure what we're seeing. And on top of that, even because we're not able to come up with the exact drug target, intracellular target that interacts to induce all these downstream phenotypes, that's been a very much of a challenge. These mice tolerate this drug incredibly well, much better than many that I've used in the past. So we know it's safe. How it works and why we're not able to see in vivo xenograft tumor harvest, some of the specifics of the mesenchymal change, that's still a mystery. And I think those are the reasons why grant proposal got triage.
Well, I think you could argue, frankly, that cancer is a moving target, and it gets these intrinsic mechanisms to counteract, so, you know, it can change its phenotype to whatever suits its survival.
Right.
So, I don't know.
A lot more work to do. Any other questions? Oh, absolutely. Thank you very much for having me.